So, Colin, Geraldine, thanks very much for inviting me to speak. And so it isn't just me today, as you said, it's Melina as well, Melina Deitch-Varela, who works with me at Moorfield. She's a junior ophthalmologist and doing a PhD in inherited retinal diseases at Moorfield, supervised by my colleague there, Professor Michelle Michaelides. And so I'm going to do the first part, try and get some excitement about cone photoreceptors, and then Melina's going to do the clever bit about how she actually measures and looks at cone photoreceptors in patients as part of her research. And so we're not really going to use slides. I want this to be a talk that people can listen to rather than see, because many in the audience, by definition, will have central visual function loss. So the slides are really just to help us navigate the talk, and we'll describe whatever is there. There's only 4 or 5 slides. And this is the second slide up now, and this just lists what we're going to cover. So I want to talk a bit about the retinal problems that involve the cone photoreceptors. So and that will have resonance to many people on the talk. What cone photoreceptors actually are, what they do, why is it that we need them, and then Melina and I will both talk about how we test them in patients. I'll say a bit about that, but I'll leave that mostly to Melina to describe. And at the end, if there's room, I might just talk a bit about genetics and why that's important, and how people perhaps can get themselves tested for their specific disorder. And we're very happy to answer any questions at the end. So what retinal problems, first of all, what retinal problems occur when the cone cells aren't working? So cones are relevant to many retinal problems, if not all retinal problems. And so sometimes, rarely, cones don't work from birth. So a person can be born without any cone function at all. And this is a class of disorders known as stationary disorders, those that don't progress, but are just there right from the beginning of life. And the general term for this is something called achromatopsia, or rod monochromatism. And these are people that are born with a genetic condition, and there are a number of different genes that can do it, that stop the cones from working. Generally speaking, they don't affect the rods. And those people have lifelong reduction in their vision, but it doesn't usually progress. So those are stationary problems, and those are probably the rarer problems we see in clinics due to cones. More commonly, there are progressive problems, and these are given different names. The names can be confusing, and that's why I tend to use the name of the gene that causes the disorder, because that's less confusing these days. But cone dystrophy and cone rod dystrophy are the terms applied to those conditions in which the cones gradually fail to work to a degree during a person's lifetime. And so a person might be born with relatively good vision, but generally then later, over years, and this varies depending upon the condition, depending upon the gene that causes it, that vision will deteriorate. Those are called cone dystrophies or cone rod dystrophies. And the most common cause of that in working age people is Stargardt disease. And so the gene that causes Stargardt disease is by far the most common cause of cone and cone rod dystrophy. But cones are important also to those people that have age-related macular disease. The reason people have loss of vision in that disorder is that the cones fail. It's not the rods that are failing or any other part of the eye. It's the cone cells that fail at the macula. And even retinitis pigmentosa patients, people may know about this class of disorders. It's somewhat opposite to macular dystrophy in that retinitis pigmentosa affects the peripheral retina and the night vision, and that's because it affects rods predominantly. But people notice a problem when, unfortunately, also the cone cells are affected. And if the cone cells weren't affected, actually, people wouldn't mind so much, because we don't tend now to use our rod vision, given that we've got electrical lighting and technology to light up our scene at night. So cones are important, and they pretty much subserve all of those functions that are missing in people that we see with retinal problems in the clinic, including AMD, age-related macular disease. So the second part of the talk is just to describe what cone photoreceptors are, what that word photoreceptor means. So cones are cells, and all living stuff is made of cells. Cells are the building block of life, and cells are basically small volumes of water and protein and DNA, which are surrounded by a protective lipid coat, and cells are specialized to do different things. So different cells in organisms will have different functions. They'll have different proteins in them and express different genes from the genome. And like other cells, photoreceptors are cells that are specialized for a particular function. And photoreceptors do many things, but they do two main things. One is they turn light into chemical energy. And so a particular protein will react to light and change its conformation and set a number of chemical reactions along the way to signal the fact that the cell has received light upon it. And then the second thing it does is turn that chemical energy into electrical energy. So there is an electrical signal that then can be sent back to the other layers of the retina and to the optic nerve to the brain, so that we can perceive the capture of that light on the retina. And just to put this in a context, the second slide, which I can describe, is just an eye, a human eye, a cross-section of a human eye, just to show where the retina is. And I'm used to using the analogy of a camera, and I'll tell people that it's like a camera in which the retina is the film of the camera. It sits at the back, inside the camera. The image is focused by the lens, which is like the cornea and the lens of our own eye. And that image then falls upon the retina and is captured with a chemical process, very much like film. Retina is cleverer than that because it then sends information back to the brain in an electrical signal. So that's the analogy I use. And on the slide is just a very simple cross-section of a human eye. It occurs to me that analogy won't last forever. And even now it's failing a bit in younger people who, many of them, never seen a camera. They just use a CCD in their iPhone. And so there'll soon come a time when I'll need another metaphor to describe what the retina is, because the camera will be an ancient piece of kit. But anyway, I'll cross that bridge when I come to it. So the cells reside in the retina, the photoreceptor cells reside in this thin layer of tissue that's at the back and communicates with the brain via the optic nerve. And the retina is a three-layered structure. So the photoreceptors comprise one of those three layers. And peculiarly, it's the layer at the back. So light has to travel through the pupil, through the jelly of the eye, the vitreous, it hits the retina, and then perversely, it has to travel through the first two layers before it hits the photoreceptor that detects it. So it's back to front is the vertebrate retina, which is a strange way to design an organ that sees, but that's the way it is in all vertebrates. So the photoreceptors pick up that signal, they do the energy transduction, turn it into chemical energy, then electrical energy, and that is signaled to the second layer, which is the bipolar cell layer, and then the third layer on the surface, the ganglion cell layer, which then is a long cell which communicates with the brain. So it goes all the way down the optic nerve, optic chiasm, and into the lateral geniculate body of the brain, the nucleus of the brain. Just a bit about how they differ from rod photoreceptors. So that photoreceptor in humans is composed of over 100 million cells, and they converge on approximately 1.5 million ganglion cells. So it's the densest layer of the retina, the three layers. And it's interesting just to reflect that the vast majority of those photoreceptors aren't cones at all. They're rod cells, rods that see in the dark. 95% of them are rods and only 5% cones. And the reason for that is that the rods are required to pick up very low levels of light. So it makes sense to have many of them converging on one ganglion cell to the brain in order to increase the chances of detecting low levels of light. And so that's why we have so many more rod photoreceptors than cone photoreceptors. And so why do we need cone photoreceptors? What good are they to the visual system? And we can think of them as doing three main things. They see color, they see movement, and they see acuity. And the reason we have two systems, first of all, rod and cone system, is that there's such a great difference in the density of light during day and night on the Earth, that one system can't do both jobs. And I don't think there's an organism that can do that with one system. So in order to have awareness of our surroundings during the night, we need this rod system, which is slow, it's magnified, but it can pick up very low levels of light. That system won't work during the day because it requires a very different set of properties. And instead, the cone system takes over. It operates in very much higher light and reacts very quickly and can see detail. So cones do those three things then. They can see color. And if we talk to people without cone vision, those people with achromatopsia who've never had cone vision, it's interesting to find out from them what it's like not to have color vision. In fact, to them, it's normal. So it's quite difficult for them to describe. And they can only do that by comparing themselves with their peers. And we can do color because there are different types of cone cells. The human retina and vertebrates differ in the number of different cone cells they have and the ability to see light. But humans have three different cone cells, red, green, and blue, and they respond to different wavelengths. And so the retina in the brain has evolved a way of integrating the signal such that it can find out to what degree it stimulates those three different cells, and so to work out what the color of the object that you're looking at is. So cones are needed for color. Rods can't do color, but cones are. They're needed for movement. So in order to see movement, that's pretty important to be able to see moving things. You need a cell type that can switch on and off quickly. Rods don't, they're sluggish. They're very sensitive, but they're slow. Cones are very fast. Not only do they switch on quickly, but they switch off quickly. So if we're trying to track a moving object, we need those properties in order to see it. And most importantly, cones do acuity, and Melina will say a bit more about this, because cones can do acuity, that is fine detail, read and to see faces and expressions, to see fine detail, because they're very densely packed. And that's interesting, and that's something we want to be able to look at directly and understand directly if we can. And so they're very densely packed, and so there's a high signal density across the retina at the center of the retina where the cones are most plentiful. And they also communicate with fewer ganglion cells. So there's pretty much a 1-to-1 ratio between cone cell and cable, the ganglion cell going to the brain, whereas with rods it's 100-to-1 or so. So they're geared towards acuity. So those three things are important to the visual system, and they're governed by cones. Just want to check how we're doing for time. Colin, am I going over?
No, you're doing okay.
I'll just finish my bit now with just how we measure the cone system in patients. So I've said that all retinal disorders, including AMD, affect people really through the cone pathway. And so we want to test them. And there are four ways clinically, four broad ways in which we can test cone function. The most basic way is to talk to the person. And many of us realize that talking to someone about their problems is a very good way of finding out what the problem is, when it's happened, to what degree it's happened, and whether it is the cone system that's causing the problem or something else, like a cataract or an optic nerve problem or a hemorrhage into the vitreous. So just talking to people, we can get a good idea about the cone system and how it's working. We can look at its structure very well now. And I've been working for 20 years in this field, and the ability for us to look at the structure of the retina is improved greatly, and Melina will tell us a bit about that. We can test its function. We can do that two ways. One, we can do it objectively without asking the patient to report anything, and that's called electrophysiology, where we shine a light at the eye and we measure its electrical output. So remember, what the eye does is a transducer. It turns light into electricity. And we can do that directly, and that's very useful to us. We put little electrodes over the eyelid to measure the electrical output, and that's called an electroretinogram. We can also do it in the brain and measure the electrical output of the brain consequent upon a flash of light. And that's called a VEP, a visually evoked potential. And then finally, we can do a psychophysical test where we can ask people to look at an object and report on it. So a field of vision is a psychophysical test. And the most important one is the acuity test, the Snellen chart. So we have in our arsenal many ways of finding out how the cone system is doing. And that's important for people like me trying to work out what's happening to someone's vision. And it's important to researchers like Melina also to understand that. So Melina, if you don't mind, I'm going to hand over to you now to say a bit about how you measure cone function in your research. Thank you.
Thank you for that. I'm sorry for the delay. Are you seeing my screen?
Perfect.
Okay, thank you. Great. Thank you, Professor Webster, for the invitation. Thank you for calling and also for having me from the Macular Society. As Professor Webster was saying, I'm going to talk a little bit about how we assess the cone function and structure. Professor Webster has just been talking a little bit about this already, but I'm just going to wrap it up by saying that indeed the first test that we use and the one that it gives us probably the most information about the cones is the visual acuity, which is the most widespread and common test used in ophthalmology probably worldwide, and not only both in the clinic and in the research facility, but also in schools and hospitals just for general screening of the vision. So as you know, after we measure how many letters the patient sees on the chart, we ruled out any other refractive error, either by glasses or pinhole, and also any other associated complications such as cataract or any other media opacity. And the remaining value usually gives us information about the cone system, particularly the one that is subfoveally, which is the one that really gives us the acuity and the three-dimension high definition vision. And once we have that number, it's been already proven that it's highly directly correlated with other structural and functional measures, such as the ones that I'm going to be talking later on. But those letters in the chart, believe it or not, it's one of the most important tests we do have to check the cone function. And a little bit further, when we're in the research setting, we also do some other testing in visual tasks, such as testing the reading speed and testing contrast sensitivity. And also another important bit for the cones particularly is testing the color vision. Of course, there's a very wide range of tests to test the color vision, going from the Ishihara pseudochromatic plates that you can find online and that combine different colors and can give us information about which of these three cones that Professor Webster mentioned is the most effective, to high-tech devices that we have in the research facility that gives you a computerized test and gives you quantitative assessment of how your color vision defect is. And again, it's a very, very thriving field where there's a lot of new tests to try to pick up these defects just by testing. So those are usually the tests that we use in function broadly in both research and clinic. And then to talk a little bit about structure, I'm sure everyone here in this call has had an optical coherence tomography, an OCT scan, which gives us an in vivo cross-sectional view of the retina. And that, as as everything in ophthalmology probably, and mainly in retina, has evolved tons over the last years. Currently, the ones we have now, the ones we have at Morpheus, for example, they can give us not only information about qualitatively how is the retina, but also quantitatively, we can measure volumes and see all the layers in depth to see if they're discontinuous, if there's, for example, a space, a gapping, or if they are disrupted, which is very nice to then correlate with all the other testings, such as color vision, visual acuity, visual field, blind spots. We can usually put everything together and look at it as a whole. And another test that I wanted to talk to you about, maybe, I don't know if you're aware of this one, is the images that I'm now showing, which is called adaptive optics technology. And it's really interesting because it's the one that is used by NASA to use their telescopes. And they use it to correct the astronomical defects caused by the turbulence of the Earth's atmosphere. And we use it on actually the same kind of purpose, because we want to, of course, image the retina on, to have these high definition images, but the imaging beam has to go through a lot of different tissues, such as the cornea, the lens, and the jelly of the eye, the vitreous. So all of these tissues cause the image to be not as defined. We lose really resolution. So by using this AO, adaptive optics technology, we can correct for all of this and have this optical resolution of 2 micrometers, where you can see each cell. And it's very important, and it's very useful when we are assessing cone dystrophies, because we mainly look at the fovea and the posterior pole of the retina, and we can see each cone set one next to the other one. And for example, it's been known that in certain conditions, such as the one that is on the top of here of my screen, that belongs to a patient with achromatopsia, which we know that their cone function and their cones in general are not working as good. And we can really see them compared to the one below, which is the normal control. We can see how the cones are wider because they have to occupy more space that was in the beginning or meant to be packed with a lot of cones. When these are not functioning very well, we see them wide. We can count them and see that there are not as many. And this is very interesting to then correlate in time, like in baseline, for example, we do, and then correlate it after a couple of years to see if it has progressed. And usually, since we do this with other testing, for example, the OCT and the letters in the chart, this gives us a very detailed information about how the disease is progressing or not. And it's also useful for treatments to test, to see if anything has changed at a cellular level after we do, for example, gene therapy in these patients. So it's a very useful test. We can see different part of the retina, different parts of the cells, such as the outer segments, the inner segments, and even the retinal pigment epithelium cells. So all of this is very helpful. And putting everything together, which is what we do in ophthalmology, putting the functional testing and the structural, we really have an idea of how the patient is seeing, and we really have an idea of how his or her cones are functioning. So we're grateful for all of you guys that are having all these tests, and this is what I wanted to tell you about.
Melina, thank you for that. So and things have changed greatly over the years. I remember a time when I started in the late 90s when we basically had a fluorescein angiogram and colour photographs, and that was it. And we had to work really hard in seeing the eye real time to get any information. So it's improved greatly, and what you're showing is remarkable. So I think we'll finish soon and get ready for questions. One thing before we do finish is to say that many of the disorders that affect the cone system, that affect the macula, and also RP that affect the cone system eventually and cause problems, are indeed genetic. And so things are easier now to find the causative gene. We're getting better at finding the cause of a person's problem. Even age-related macular disease is a genetic disorder, and a very well understood one, but it's due to many different genes all having a small effect on the person. Whereas younger people with Stargardt disease, for instance, or cone rod dystrophy, or even RP, usually have one gene that's causing the problem. So we are getting better at it. The NHS is gearing up now to doing more testing in people with more extensive testing in people with these conditions. And so it's a reasonable thing to do if you don't know what causes your eye condition, to at least get a referral to an ophthalmologist and to begin that journey, which is a shorter journey than it used to be. We still only find the genetic cause in about 50 to 60% of people overall, but that's improving. And the more people we test, the more tests we do, the better it will get. And that helps all of our research. It certainly helps the research that Melina is doing when she's looking at specific disorders that are pretty much defined by the gene that causes them. Colin and Geraldine, I think we've finished our bit now, and we're very happy to answer questions if there's time.
I think there's plenty of time, thanks. Really interesting, both of you. Thanks very much for explaining it so clearly. I love these things. I would learn so much. It's brilliant. So Geraldine, have we got any questions?
No, not yet. I've got a few questions while people have a chance to write things in the chat function. I've always been a bit confused about cone dystrophy, rod cone, cone rod. Can you say anything about that? Are there really differences in those, when people are given those diagnoses, are there really differences between rod cone and cone rod? I've never quite understood that.
No, you're not alone. These are old-fashioned terms before we knew the genetic cause. So nowadays we tend to, whenever we can, describe a retinal problem, a retinal dystrophy. So retinal dystrophy just means a retinal problem that's likely due to a gene. We tend to describe it in terms of the gene that causes it. But roughly speaking, a cone dystrophy is one that most people, this will differ. In the States, they tend to use this differently. But in Europe, it's usually reserved for a progressive problem with the cone system. That is not a problem someone's born with, but something that occurs. And forms of Stargardt disease do that. Cone dystrophy tends to mean it affects the cones and not much the rods. Cone rod dystrophy means it affects the cones and the rods, but the cones more. And rod cone dystrophy, which is pretty much synonymous with RP, retinitis pigmentosa, means that it affects both the rods and cones, but the rods a bit more, because that's at the front of the word. It's confusing. And well, one of the problems, I've met a few people that have got upset by a change of term. So they might have been called a cone dystrophy, and I might write in the letter that it's a retinal dystrophy because it's both rods and cones, and I don't want to prejudice either class of photoreceptor. And they write back to me saying, I've got a different condition now. One I've always thought I had is not the case. People mustn't worry too much about the names because they are changing and evolving. But at least now we understand the disorders better, and that's exemplified by the fact that we can often refer to the gene that causes them. And very often the specific spelling mistakes. So in Stargardt disease, it isn't just a gene, it's the precise changes that we understand and give rise to the different manifestations of the problem.
Thank you.
I don't know whether that helps, Jodie.
Yeah, well, when I always say to people, it's the genetic diagnosis that is more important than the name, because the names were given at a time when we didn't have the genetic diagnosis, and the genetic diagnosis is more precise. So I try and explain it that way. Okay, we've got our first question. Excellent. Is gene CACNA2D4 associated with any specific cone dystrophy? And is this a common gene to find dysfunction? I've not heard of that gene before.
Melina, do you want to do that one?
I was about to look at the gene yeah.
Well, I know about this. So it's actually a rare gene, and so the question asks about a gene called CACN-A2D4, and CACN stands for calcium channel. Ca is calcium; it's the elemental symbol for calcium, and Ca is meant to stand for channel. So it's a gene that makes a calcium channel. And calcium channels are important. They exist in photoreceptors, and they couple the electrical activity of the photoreceptor to the signal across the synapse to the next layer of the eye, which you'll remember is the bipolar cell. And so that coupling is achieved through calcium channels, and that particular gene makes part of the calcium channel that performs that trick in all photoreceptors, both rods and cones. And so that gene indeed does cause cone dysfunction, but it also causes rod dysfunction. So it's not a pure cone dystrophy. And we're still unsure as to whether or not it progresses much. So this particular gene might cause a stationary disorder in which people are born with problems, or at least they develop problems that then don't progress any further. So it's a very interesting gene, and colleagues of mine, such as Professor Omar Maru, who works at Moorfields, is particularly interested in the electrophysiology of the eye, the way that electrical signal is made in response to light. And so if there are people out there with CACN A2D4, he'd love to meet you.
Lovely, thank you. We know Omar Maru. He got one of our Macular Society awards a couple of years ago, so we can put you in touch. So the next question is, I have BEST1, I think that's what that says, picked up at early 30s by myself, despite always being under eye hospitals for checks. I'm long-sighted all my life and have good colour vision still at 74, but noticing slight problems with pale shades of grey, beige, yellow. I've been lucky so far. That's not a specific question, but I would want to comment.
So Best disease is a problem that affects the macula, and it's a variable one. And I do see, I see people presenting as a young person with loss of vision. So it can have a detrimental effect on central vision, and in some unlucky people that can affect both eyes. But most can inherit the same gene and do pretty well through life. They either have good vision in one or both eyes that can last until at least the 8th decade, like this person, and of course beyond. So Best disease can be quite benign in some people, yes. And if you notice changes, then of course it's worth just being checked, if not already, to make sure it isn't anything else like cataract that's affecting the condition. And very rarely, Best disease can be associated with little blood vessels growing. And so we're always a bit nervous if someone has a sudden change in their vision, because we'd want to check that isn't the case, and if it is, to treat as we would treat AMD. But yes, it is quite benign, and there are a number of people that are lucky with Best disease. And one of the aims of research is to find out what the lucky people have that the unlucky people don't have. So why is it that only certain people lose their vision when they inherit the gene for Best disease? And some families will know this when they look around the families and how different people are. And so there are reasons for that. And there are likely to be other genes that cause that luck. And we haven't found them yet, and we'd like to. So part of our research at Moorfields, and this is a larger study in the country run by the NIHR, National Institute of Health Research, which is NHS funded research. We're trying to address that problem by collecting together as many people as possible with those conditions. Thank you.
Thank you. Somebody's asked for Omar's name and number. I don't have a chance to access that now, but I'm sure we could. Do you know? Would you mind?
Sorry, not at all. So Maru is spelled M-A-H-R-O-O. That's Omar's surname. And address any query to Moorfields, either by post or by phone, and it'll get to him.
Thank you, Milena, for putting his name up there. Okay, we don't have any other specific questions. Andrew, we've just started funding a project with you on the ABCA4 gene. Would you like to explain a little bit about what that project involves? Because it is mining data.
It is, and so thanks very much for that. And one of the challenges we have now in macular disease and rare disorders is being able to manage large data sets. So we're lucky, particularly in the UK, in that we have a lot of data on patients with rare disorders, not just eye disorders, but other neurological disorders, cardiology disorders. And so we need people who have the ability and the training to get information from large data. And that's quite rare, but we do need those, and we need to fund them to do it. And so at the moment, we've got a lot of data from the National Institute of Health Research. We've got a lot of data from the Genomics England project. So that recruited over 50,000 families with rare disease, and a significant proportion of them have eye disease, because ophthalmologists around the country were very quick to recruit to that study, partly because we understand the genes that cause problems with the eye for numerous reasons. So we've got this great data set to work on, and increasingly now the NHS is going to be adding to that. So whenever someone has a genetic test, they're given the option of being included in that research project. So it's going to grow. And so the aim of our project is to look specifically at the most common gene that causes Cohen dystrophy and macular dystrophy, and that's the ABCA4 gene. Because there are many things we don't understand about it. And if we were to understand them, I think we'd treat people better, and we'd aim treatments better and have more logical trial design. And so there's an interest in the specific variants that cause the disease in the ABCA4 gene. And to give an example of one interesting question is that some spelling mistakes that we see in our patients are also seen out there in the general population, seemingly benign. They're quite common in the general population. They don't cause Stargardt disease in the majority of people, but in other people they do. And so we're trying to work out why, and whether it is something else that's on the same chromosome that has an effect on the spelling mistake itself, or whether it's the other chromosome that might have an effect, or a different gene interfering with the ABCA4 gene. So to answer those questions, we need to mine the data, and that's what we're going to do. We're joining up with others. So this is a worldwide consortium. So we're joined up with people in Europe and people in the States, so we can share data. And they have their own people, genetic epidemiologists, looking at the data too. So hopefully that will be fruitful, and that's a study that certainly needs doing. Thank you.
Thank you. Okay, a couple of other questions have come in. From Leslie, my 21-year-old son has Cohen dystrophy and his eyesight is deteriorating. Can I ask about trials and some information on recent trials that may have been done?
Melina, do you want to ask?
Sure. Yes, thank you, Leslie, for the question. I think it's very important what Professor Webster said at the end of his talk about the genetic cause of your son, in this case, Cohen dystrophy. There are a lot of trials, and there we are really doing a lot of different approaches for the genetic conditions in the eye, but usually we need the genetic confirmation of all of them. So without that information, it's really hard to really assess you and what opportunities are there for him. Once we know the gene, we can really talk about what's out there, either here or abroad, for him, and what things are also in the preclinical stage that we're developing. But once that the genetic testing is done, I'm sure we can give you more information. But just as a broad picture, there's, for example, for Stargardt disease, I have no details. Okay, but from what I understand from your answer, it's going, isn't it? It's on the process. So, for example, if it's Stargardt, what is the most common thing? There are some trials that are, with pharmacotherapies, which is oral drugs. There's also gene therapy, gene editing techniques, and a lot of other things for other genes. But again, once we know that information, you're probably going to have to do this question again, and we'll answer it with more information.
Thank you. I do advise people to get a genetic diagnosis if they can, even if they were diagnosed with a dystrophy some time ago. Go back and ask for a genetic test, because it's becoming a lot more easy to access them.
Yeah.
Just to add to that, just to emphasise, I'm sorry to pour water on things, but there are no treatments yet, so people aren't missing out anywhere with the treatment, and also trials aren't treatments. at all. And so to enter a trial is an altruistic act by a person with a condition who isn't getting a treatment. They're adding to data, and they're most likely getting something that won't in the end be a treatment, but they're helping reach one. So it's an act that we're very grateful for, but it's not a benefit to the person. And so people mustn't think they're going to miss out on a trial because they're in the wrong part of Britain, or they don't know what their gene is, or that we don't need any more participants. And very often we don't. There's plenty of people that participate in these. So don't worry if you're not part of the trial. One strategy is to see which works, and then you'll want the treatment. And then that's when we worry, because we'll want to make sure the treatment gets to all of those people with the disorder who would benefit from it. And that, fortunately, we do have that dilemma with one condition. It doesn't cause macular disease, it causes a whole retinal disorder, Leber's congenital amaurosis, and that's RPE65. So it's a great problem to have. We're now needing to find folks who've got that condition so that we can treat them with a drug that works. This is gene therapy that we hope will prevent further visual loss, and in some might bring back vision. So we'd like a few more of those challenges in the future, and hopefully we will before at least I retire in a few years' time.
Thank you. Okay, Evangeline has asked, can a drug toxicity cause retinal dystrophy, or is retinal dystrophy caused only by gene mutations?
So that's a very good question, and yes, it can. So drugs can mimic retinal dystrophies, and so all of us working in this area should, and we hope we do, always ask about a drug history. What drugs have you been on? What drugs are you on? And there are certain drugs we know of that cause problems. Chloroquine and hydroxychloroquine are the classic ones that can cause problems with the central retina. But there are others, and we might learn of new ones. We always ask. So yes, that's something we always ask about. And apart from genes and drugs, if you've got a symmetrical change in the eye, there aren't many other things that can cause it. So we always do ask that, yes.
Thank you. And Ellen asks if you know Dr. Stephen Tsang, T-S-A-N-G, from America.
So I do, yes. Yes, he used to work at Moorfields. He's a brilliant clinician and scientist, and works at Columbia now, yeah. And we collaborate with him too. So I think we've just signed a, collaboration is hard. We'd like to collaborate with people more often than we do, and there are lots of documents involved and letters, and it's really quite difficult, unfortunately, to collaborate with colleagues elsewhere. But we do it whenever we can, and Stephen is a very collaborative clinical scientist in Columbia, New York. Yes. Was there a reason specifically, Ellen?
The person said it's her doctor, which is, so she sounds like she's a very lucky person. Yeah. That's praise indeed. So that's excellent. I'm aware of time. So if we have any more questions that come through the chat, if I can send them to you, Peter and Melina. Okay, everyone's name's wrong. Andrea and Melina. Tonight, I will. If you wouldn't mind answering them that way, that would be very kind of you. If I get your name wrong again, of course, you can throw me out the window. We'll never speak again. But I don't know why I'm getting people's names. Moving on. So, Jerry, that's sort of leave a little bit of time for you just to talk about a couple of bits before I talk about a couple of bits. So, just to say thank you to Andrew and Melina, and then over to you, Jerry, for a second or three.
Thank you. Just in case people may not be familiar, but be coming to the Macular Society quite recently, we place a great deal of value on funding medical research to find a cure for all macular diseases. And we have a research program. We fund projects as like the one we are funding with Professor Webster. Over £1 million worth of research projects we fund every year, and we're planning on growing that research portfolio in coming years as our income grows. We also spend quite a lot of time devoted to what's called patient and public involvement in research. We have something called our research participant database, which is a way that people can sign up and let us know that they would like to hear about research opportunities. And it's not just about being part of clinical trials. There's lots of other ways that people can take part in research. Any trial, any study research really needs to involve patients these days to advise on the research. The patient's point of view in research is really important these days. So we're always looking for people who are happy to take part in research, either maybe commenting on the patient information materials, making sure that they're clear and understandable, to be on focus groups, patient advisory boards, lots of opportunities that are potentially available for people not just taking part in clinical trials. So if you're interested in that, on our website in the research section, you can sign up to the research participant database. There's a whole page devoted to it with a form. We don't require very much information from you. Really, we just need to know what you would like to do, where you live, how far you like to travel, because you probably always need to attend a hospital or a university. And then we keep you on our database, and if opportunities arise, we'll be in touch. So do have a look at that, think about whether you might like to volunteer. It's a volunteering opportunity to be involved in research in some way or other. Thank you very much.
Perfect, thank you. Lots and lots going on. We get contacted a lot, it's fantastic. Which leads us on beautifully to our annual conference. So last year, because of COVID, we had a virtual conference, it was online, and this year we decided to do the same thing again, because last year it was amazing, really. We had about 1,600 people throughout the day looking at our conference. So this year it's online again, and it's on September the 11th. So the inherited retinal disease section starts in the morning at 9:30, he says, struggling to remember the times. And the AMD sessions, the age-related macular degeneration sessions, are after lunch. But throughout the day this year as well, we've got some additional sessions around day-to-day living with macular disease and well-being and things like that. You can get loads of information about the schedule and the program on the Macular Society website. So if you just Google Macular Society Conference 2021, it will pop up, and it's free, which is fantastic. So once you're there, you may as well just register for the sessions, and you can dip in and dip out all day to your heart's content, basically. That's some really fantastic speakers talking about all sorts of different conditions, and we've also got somebody from British Blind Sport, and we've even got a member of Paralympic Team GB come to do a bit of a chat as well. So something for everyone, I think. So just leads me to say that next month's virtual clinic is going to be a bit different, because obviously we've got lots of research conversations and conditions conversations and discussions coming up because of the our virtual conference. We're going to have Sight and Sound, who are going to come along and talk about technology and some of all this clever equipment that people use to get around their difficulties with vision on the computers and magnification and things like that. So that's going to be an interesting conversation. And that's on the 30th of September. And again, it's at 6:30. So just keep an eye on the old Macular Society website, and you can watch to register. And if you fancy watching any of our previous virtual clinics, they're on there too. So there's one on Best disease. I know someone mentioned Best a little earlier. So there is Professor Andrew Lotry did a session on Best disease for us a wee while ago. So that's on there. It might be worth a watch. And so finally, that's kind of us for the evening. So thanks again to Professor Andrew Webster and Melina Dutch Varela. I nearly got it right eventually. Terrible problem with names today. I do apologise. And if we don't see you at the annual conference on September the 11th, hopefully we'll see you at another virtual clinic at some point fairly soon. So thanks very much.